Hello, everyone, and welcome to today's webinar, Unveiling Novel Biomarkers and Their Spatial Distribution Within the Tumor Microenvironment of Renal Cell Carcinoma, using the BondRx and image analysis workflow. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Now, we encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Additionally, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. I would like to bring your attention to a survey box on the far left of your screen. We'd love to see your answers and ensure that we can assist you with all of your questions and automated staining needs. Now, I would like to welcome our speaker, Dr. Nourhan Ahmar, post-doctoral research fellow, Brigham and Women's Hospital. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. Dr. Ahmar, welcome. It's so wonderful to have you today. You may now begin your presentation.
Thank you so much for the introduction. So as mentioned, today I'm going to talk about how we can use the BondRx and the image analysis workflow in order to know better about the tumor microenvironment of renal cell carcinoma. So this is my outline. First of all, I'm going to give a bit of background about our workflow, and then I'm going to talk about the workflow and how we optimize it. And then I'm going to provide two applications of how we use this workflow in our research, and finally, the concluding remarks. So why are we interested in renal cell carcinoma? Renal cell carcinoma tumor cells are very unique, by which they have missense somatic mutations of the host genes that will produce neoantigens. These neoantigens are considered to be foreigners, and therefore they will trigger a host immune response in a process called the cancer immunity cycle. The neoantigens will be recognized by the dendritic cells around this area, and therefore the dendritic cells will carry these neoantigens on their surface, and they will take them to the tumor draining lymph nodes, by which they will activate the naive CD8 positive T cells. Then after that, the CD8 positive T cells recruited into the tumor area will start to reactivate something called the tumor experienced CD8 positive T cells, and they will start attacking the tumor. After that, we will have a tumor lysis that will release additional neoantigens, driving this cycle again. And this process basically will provide a complex tumor microenvironment that is very rich and has a heterogeneous population of immune cells. And different subtypes have different immune microenvironment, making renal cell carcinoma, in specific clear cell renal cell carcinoma, the most highly infiltrated solid tumor. Now, what about the tumor microenvironment and the treatment of renal cell carcinoma? So the treatment of RCC has evolved over the past decade. After the widespread use of VEGF tyrosine kinase inhibitors, immune checkpoint inhibitor came and they revolutionized the treatment of RCC. That's why it's very important to understand the tumor microenvironment with all the immune cell population in it, because this TME plays a critical role in cancer progression and response to immunotherapy, specifically in ccRCC. So here comes how the finding of predictive biomarkers come to place. The first-line treatment of advanced clear cell renal cell carcinoma today, it includes tyrosine kinase inhibitor plus immune checkpoint inhibitor, or a dual immune checkpoint inhibitor. However, with all these technologies and treatments, complete response is still rare. Here comes the urgency to identify reliable biomarkers that can predict clinical outcomes to immune checkpoint inhibitor therapy. And this will aid oncologists in stratifying patients and tailoring targeted therapy on a personalized basis. This is just a simplified slide of the complex tumor microenvironment that includes so many immune populations. And actually, I see it as if it's a dynamic battlefield we have some immune cells that are going to fight with the tumor and allow for tumor progression. An example on that would be the pro-tumoral macrophages. They are called M2 macrophages. And then we have those that are fighting against the tumor, and they allow for tumor suppression. And these include macrophages that are anti-tumoral, or M1 macrophages. Now tertiary lymphoid structures also have been gaining interest in research. We have also the CD8 positive T cells, the cytotoxic T cells. Now, this is a summary of our workflow in the lab. We use multiplex immunofluorescence and image analysis assays in order to know better the tumor microenvironment by targeting specific biomarkers in it. So we use the BondRx, which is our automated workflow, in order to stain unstained slides. And this is from the Leica Biosystems. And then after staining with the multiplex IF, we go into the PhenoImager HT by Akoya in order to have a scanning of digitally scanning the slides. We view the slides or the images via the PhenoChart software. And then after that, we take hotspots or regions of interest from the tumor in order to view them on Inform and do the unmixing of these specific wavelength into their channels. And then we use the halo from Indica Lab in order to use or to quantify and phenotype the cell population, and also do spatial analysis on that. This is a detailed slide of our workflow. In our lab, we receive formalin-fixed paraffin embedded blocks or unstained FFPE slides from patients that have advanced clear cell RCC enrolled in multiple clinical trials. And then after choosing the assays or the assay or the biomarker in that assay, we go and we stain those slides with a chromogenic IHC. And then after that, we shift into a multiplex IF assay that includes that uses mainly the opal fluor tyramide-based system using the BondRx. And then after that, as I mentioned, we scan the slides using the PhenoImager HT, and then we view the slides, we take hotspots, we do the unmixing using Inform, and then after that, we take the slides or the images into the halo in order to do the analysis and the interpretation and the quantification, and also study the spatial distribution of these biomarkers using the spatial analysis algorithms. Here I'm going to give you an example on how we optimize a multiplex IF panel, and I'm going to use a real example from one of the panels that we optimized. So here, the first step is choosing the biomarkers that you are interested in targeting in the tumor microenvironment or investigating in the tumor microenvironment. And in our case, we had these 6 markers. We were interested in studying the CD4 and FOXP3. They are the regulatory T cells. And we were specifically interested in studying the PD1 expression on those cells. So we had CD4, FOXP3, PD1. We were also interested in studying the B-cell mediated structures. They are called tertiary lymphoid structures, and they express CD20, CD3, and CD21. So first step is choosing our biomarkers. And then the second step would be choosing reliable monoclonal antibodies for these biomarkers. And then the third step would be choosing a control. In our case, we chose a high-grade inflamed clear cell renal cell carcinoma that that we know that this tissue expressed those biomarkers in high levels. And then the fourth step is going to be determining the primary antibody dilutions using chromogenic IHC. So basically optimizing using chromogenic IHC. Once we have for each antibody the initial dilution, and we know that we are satisfied with the chromogenic IHC, we then move and switch into multiplex IF. First of all, we start with a monoplex IF before we do the monoplex IF, we need to sit down and put like a chart or a plan of how are we going to match the antibodies with the opal floors. And I'm going to provide an example of how we did that in this panel in the later slides. Then we are going to optimize the primary antibodies conditions and the opal floors dilutions in a monoplex IF for each specific antibody. Then after that, we are going to compare the single IHC chromogenic with the monoplex IF by choosing a specific area from the IHC and from the monoplex IF to ensure that we have the same count and same intensity. That's why the best thing to do is when you are cutting the control is to do serial sections so that you can compare exactly the same area. And after we're satisfied with each monoplex IF, we go and combine the markers into a sextuplex. We also make sure that each specific marker on data channel in the multiplex matches that of the monoplex. So also we take a specific area and we compare each channel with from the multiplex to the monoplex. And then the final step, after we ensure that we have a multiplex IF panel ready, we do something called quality assessment and the drop control that I'm also going to discuss in detail in the later slides. So this is a slide by which I'm providing the reagents that we used in optimizing this specific panel and all our multiplex IF panels. We use the BondRx research detection system from Leica. Also, we use the Bond protocol Opal 7 color version 5.2 plus, and this protocol is very compatible with the Opal tyramide system floor from Akoya. And then in this specific panel, I used these floors, the Opal 480, the 520, 570, 620, 690, and the 780. We used DMSO in order to reconstitute these floors, and then we used DAPI. We used the automation amplification diluent in order to, which is the diluent for the floors. We use the antibody diluent to block and the polymer from Akoya that has anti-mouse plus anti-rabbit HRP. In this panel, our antigen retrieval was ER1 for 30 minutes. And also sometimes we replace some reagents, that's why I like this protocol, because it's very dynamic by which you can add and remove reagents as much as you want. We use here the Leica Novocastra post-primary from Leica and the polymer also from Leica, and I'm going to explain how we integrated them into the Bond protocol. So this is how the panel looked like, and each antibody was linked to a floor, and also the position here matters just because of the repeated antigen retrieval is going to affect the intensity of the floors. So here I'm going to provide the rationale of how we match these antibodies into with these specific floors and this specific position. And definitely optimizing a multiplex IF isn't something easy. It's definitely you will have some challenges. And I'm going here to talk about some of the issues that we faced and how we were able to overcome them. So first of all, I will start with the anti-CD4. This antibody was a bit challenging. It was the hardest antibody to optimize because there are multiple expression for this antibody. It's not only expressed on CD4, but also some macrophages capture this antibody. So that's why it was placed in the first position. It will allow me to optimize it in a better way and optimize it based directly on the IHC without being affected by multiple heating steps. So I placed it with the Opal 620 in the first position because Opal 620, as you can see here, it has the least overlap with other floors in terms of wavelength and has the least background. So anti-CD4 antibody, whenever I was optimizing it, it was very weak with IF, but it was normal with IHC. So that's why I had to increase the concentration of the antibody and the fluor, but it's still very weak and not resembling the IHC chromogenic optimized one. That's why I had to incorporate an amplification step, which is adding the post-primary from Leica. It happened to be a rabbit anti-mouse, and this antibody is a mouse. So this is a way whenever you have an antibody that is mouse and it's very weak, you can add this post-primary as an extra amplification step, and it actually worked beautifully for the anti-CD4. And then we had the anti-CD21 and anti-CD20. These two were placed with the Opal 480 and 520. They are rare markers in general, and they were placed with the highest intensity fluors. Basically, just one note here when using the Opal 480 and 520, we noticed that it overlaps a lot with autofluorescence wavelength. So sometimes when you put markers with Opal 480 and 520 and you're doing unmixing, you will find that sometimes this marker the intensity will drop after unmixing, because you know here that you need to increase or amplify the antibody step. So to overcome the disappearance of these markers after unmixing, you need to increase the dilutions or the intensity of the markers placed on those fluors. Finally, the FOXP3. The FOXP3 is a nuclear marker, and thus it requires a very harsh antigen retrieval. That's why it was placed in the last position. It will benefit from the extra heating steps. So that's why we always place nuclear markers on the last step. And it was placed with the Opal 780. And here, for some reason, and with trial and error, when I replaced the polymer of the Akoya with the polymer of Leica, I was able to find better signal or an improved intensity. And it's just something based on trial and error. So this is basically a summary of the rationale of how we matched the antibodies with the fluors and some of the issues that we faced while we are doing this panel. And here I'm going to talk about the drop control, which is basically a quality assessment step that we do to check the stripping between each reaction. And in our case, we had 6 markers or 6 steps. That's why we want to do a 5 sequential multiplex IF stains using 5 sections. So in the first stain, we are going to keep the first reaction by adding the first marker and the first opal, and then we are going to drop the second marker and keep the second opal. By this step, we are going to see if there is any bleed through of the first marker into the second opal, and we ensure that the first reaction has been stripped properly. So that's why we do this sequentially for five stains in order to make sure that each step was stripped properly. Now I'm going to talk about 2 applications of this workflow in our research. Application number 1 is going to be the IF biomarker, and application number 2 is going to be the regulatory T cells. These are practical and the clinical examples that we use in our lab. The first one is the IF biomarker. So I'm going to provide a bit of background. We know that the cytotoxic CD8 positive T cells are crucial for recognizing and killing tumor cells. And we have here, I'm going to talk about 3 immune checkpoint receptors that are expressed on the T cells, the PD1, the TIM3, and the LAG3. And the CD8 undergo changes in the tumor microenvironment, and whenever the CD8 express PD1 but does not express TIM3 and LAG3, they are called antigen experienced but non-terminally exhausted T cells or partially exhausted T cells. And whenever the CD8 positive T cell express PD1, TIM3, and LAG3 or LAG3 is considered to be terminally exhausted, and therefore they lose their effector function and become dysfunctional in the tumor microenvironment. So based on that, we hypothesized that we wanted basically to see what is the effect of these cells in patients that are taking anti-PD-1 monotherapy. So we hypothesized that patients that have, of course, with advanced clear cell renal cell carcinoma that have high levels of the CD8 positive tumor infiltrating lymphocytes that are expressing PD-1 but negative for TIM3 and LAG3 have better response, because upon anti-PD-1 monotherapy, they will be regenerated into functional cytotoxic CD8 positive T cells. However, those that are terminally exhausted will not be able to be regenerated upon anti-PD-1 monotherapy. And in order to study this hypothesis, we optimized a multiplex IF that includes CD8, PD1, TIM3, and LAG3. And this is an example of the multiplex that we optimized. And here we see that we have an example of patient number 1 that has CD8 positive, PD1 positive, but negative for TIM3 and LAG3. And here another example of a patient that has CD8 positive, PD1 positive, TIM3 positive, and LAG3 positive. And of course, we used image analysis to quantify these markers. And here we stained samples from the tissue samples from the HCRN GU16 to 60 clinical trial. We stained 67 patients that have advanced CCRCC, and they are taking first-line nivolumab therapy. And we were able to prove that patients that have high levels of this specific biomarker, which is CD8 positive, PD1 positive, but negative for TIM3 and LAG3, and for short, we called it the IF biomarker. These patients that have high of the IF biomarker had higher ORR and longer progression-free survival. So they are basically doing better. And when we tried to combine this biomarker, the IF biomarker, with another biomarker, which is the PD-L1 expression on tumor cells, we were able to find out that patients that have high IF biomarker and have positive PD-L1 expression on tumor cells were actually doing the best. And they had 75% objective response rate and 24.7 months of median PFS. And then our conclusion for this biomarker was that high levels of the CD8 positive, PD1 positive, TIM3 negative, LAG3 negative tumor infiltrating lymphocytes are associated with improved response on nivolumab or anti-PD1 monotherapy. And we were able to prove this theory in 3 independent clinical trials. We proved it in the Checkmate 010, in the Checkmate 025, and in the HCRN GU16 to 60 clinical trial. The second application is the regulatory T cells, which is another component of the immune microenvironment. And we were specifically interested in the PD-1 expressing regulatory T cells. So the aim of studying this was because many patients still do not benefit from anti-PD-1 monotherapy, and they are actually resistant to nivolumab or anti-PD-1 monotherapy. So that's why we think that there's some mechanism in the regulatory T cells, specifically the ones expressing PD-1, could be a predictive biomarker of resistance. So normally in our normal state, the regulatory T cells, which is CD4 positive, FOXP3 positive expressing cells, they are there in order to balance our immunity and suppress immunity whenever it's needed and decrease the CD8 positive effector T cells. However, when these regulatory T cells are expressing PD-1, they will have a reverse function, just because PD-1 on any cell is considered to be inhibitory, or it inhibits the function of this cell. However, in the context of anti-PD-1 monotherapy, we think that the anti-PD-1 will inhibit the inhibition of these cells and therefore suppress immunity again, causing resistance to nivolumab. So we think that PD-1 expression on tumor infiltrating Tregs is a biomarker of resistance to anti-PD-1 monotherapy. In order to study this hypothesis, we optimized a 3-plex that contains CD4, FOXP3, and PD1, and we quantified the PD1 expression on the CD4 plus FOXP3 positive cells. And we were able to study this hypothesis in the Checkmate 025 clinical trial. And we were able to actually conclude that patients that have high expression of the PD1 on tumor-infiltrating Tregs were associated with resistance to anti-PD1 nivolumab, as we see here in this PFS graph. And also the OS was associated with they basically have lower overall survival than patients that are on everolimus, where we didn't see any relationship between this biomarker. So it's very specific to nivolumab therapy. So here I talked about first the IF biomarker, which is a biomarker of response to anti-PD1 monotherapy. I also provide an example of the PD1 positive Tregs that is a biomarker of resistance to anti-PD1 monotherapy, as we proved it in this trial. Now we thought about creating a model that integrates those two biomarkers together. We called it the ratio of the Treg positive PD1 over CD8 positive, PD1 positive, TIM3 negative, LAG3 negative. And our statistics showed that this model actually had a higher predictive value than each marker alone. So everything I talked about can be summarized in this balance, where each patient we can assess if they have high CD8 levels of CD8 positive PD1 positive, TIM3 negative, LAG3 negative effector T cells. We know that they are going to benefit from nivolumab, and they will have an enhanced anti-tumor immunity. However, if the patient has high PD1 positive Tregs, we know that they will not benefit from nivolumab, and additional treatment or other treatment should be added in order for this patient to benefit from it. So also we studied the spatial distribution and the interaction between these biomarkers in the tumor microenvironment using the spatial analysis nearest neighbor software. For that, we did on one tissue section the both biomarkers. So we had to optimize a multiplex IF that included the IF biomarker with the regulatory T cells. And we measured actually the distance between different Tregs and different exhausted states of the CD8 positive T cells in order to maybe try to hypothesize that the regulatory T cells, whenever they come in contact or close to the CD8, they will render them as terminally exhausted. So we hypothesized that maybe there is a shortened distance between terminally exhausted T cells and the regulatory T cells, but our analysis did not prove this hypothesis. So finally, using the multiplex IF and the image analysis, and using the automated workflow by Leica Biosystem, we were able to have a closer look at the tumor microenvironment in renal cell carcinoma, and we came into these two clinically relevant conclusions: that patients with high levels of CD8 positive, PD1 positive, TIM3 negative, LAG3 negative tumor-infiltrating lymphocytes, what we call the IF biomarker, was associated with response to nivolumab monotherapy in patients with advanced clear cell renal cell carcinoma in 3 independent clinical trial cohorts. As well, we were able to prove that patients that have high percentage or levels of the Treg positive, PD1 positive T cells are associated with resistance to PD1 blockade in advanced clear cell renal cell carcinoma. Finally, I would like to thank my mentor, Dr. Sabina Signoretti, and my lab members. This work was truly collaborative. And thank you. Now, I would like to take some questions.
Dr. Ahmar, thank you so much for your important research that you're sharing with us in your presentation. This has been so great. It is now time for our live Q&A portion of our webinar. Now, to our audience, if you haven't submitted any questions yet, please do so now. Simply type them into the Ask a Question box, click Send, and we will answer as many of your questions as we have time for. So, Dr. Ahmar, we have some great questions that have already come in. But before I dive into that first question, I just want to remind our audience that we have a great survey on the far left of your screen. We'd love your participation, and we'd love to make sure that we are able to answer all of your questions. So, Dr. Ahmar, let's start with this first question here. What is the typical timeline from the initial brainstorming of the biomarkers panel to completing the staining, scanning, and image analysis process?
Yeah, can you hear me?
Yes.
Yeah, so I think this depends a lot on whether you have the Bond RX availability in your lab. So the first time I personally did this panel from beginning to an end, we didn't have the Bond RX. We were using it only on the weekend. So it took some time. It took around 6 months between optimizing the panel, making sure of the quality control, and then doing scanning, because also this requires another scanning machine, and then the image analysis, of course, depending also on how many slides you did. However, whenever we had the Bond RX available in our lab, the second panel took us like 3 months in total to do it, to do the optimization, to go over the scanning and also the image analysis, and we were able to get results very quick. So I think having the Bond RX is very important in the lab, because first of all, it's a quicker way and very consistent, and it allows you to do 30 slides at a time. So the more you do, the faster you will be able to do your panel.
Thank you. Our next question. How easily accessible is the software? How can it be used to effectively integrate AI models into this workflow?
Yeah, so after staining the slides, we perform multispectral scanning, followed by quantification of immune cells using softwares, image analysis softwares. So in our lab, we have the Halo and the Halo AI from Indica Lab. However, there's also free resources that other labs can also rely on, which is the QPath. That has also an integrated AI in it. Although it's not that useful, user-friendly as the Halo, however, there are so many tutorials that someone can rely on. Another one that was very beneficial for the segmentation was CellPose, that also has an AI feature in it. So these are also free resources that can be integrated to this workflow easily.
Very good, thank you. Our next question, and this question's two parts. What criteria was used to identify the region of analysis, and how was the tissue area annotation performed?
Yeah, so the region of analysis in these two examples that I provided were intratumoral. So basically, we had one tissue section from a nephrectomy of 67 patients in our case. And each time we do the staining and then the scanning of the whole tissue scanning, and then we take hotspots or regions of interest using the Inform software. And then after that, we do unmixing. And then we do the quantification. And how was the tissue area annotation performed? As I mentioned, it is intratumoral, and it was actually through PhenoChart and then Inform. It will allow you to take the specific area inside the tumor, because we're interested in what is happening inside the tumor, not at the periphery or in the normal kidney, but it also depends on your aim of studying or the aim of your research.
Very good, thank you. And our next question, this one is also two parts. Do you still rely on manual IHC staining in the lab? And if so, what challenges do you encounter when transitioning from manual methods to automated platforms like the Bond RX?
Yeah, so
We very rarely rely on manual IHC, to be honest, after we have the BondRx. As I said before, the BondRx is quicker and more consistent, especially when it comes to IF. But we do have, very rarely, we optimize very challenging antibodies using manual IHC. The main thing about these two is that the automated RX staining relies on a polymer-based detection system, and our manual IHC protocol, and I know there are plenty of manual IHC protocols out there, but our manual IHC has also a polymer-based detection system. So whenever we are transitioning between manual to automated or vice versa, we do not really change a lot in the starting dilutions, just because the detection system is the same. But in general, we definitely shifted from automated, especially the IF, because it's very fast. We can do so many chromogenic single IHCs per day. And if we want to do a long multiplex, we do it overnight. Like you can do basically every day. Instead of manual, it's going to take just one stain the full day.
Very good. Dr. Ahmar, thank you so much for sharing all of your experience and your expertise with our audience. Do you have any final comments for our audience?
Maybe I can share a very useful resource that during my postdoc, I relied a lot on when I'm doing the optimization. Should I drop it somewhere, or is this available? Drop it?
Yes, if you want to just put it in the chat real quick, we'll send it out for to our audience. Yes.
Okay, sounds good. I will do that right now.
Sounds good. Yes. So while we're waiting for that resource for you, I just want to make sure everybody remembers to fill out that survey on the far left of your screen. And you'll also notice in the bottom right corner of your screen, Dr. Ahmar's resource has popped up for you down there. So thank you for that. So Dr. Ahmar, thank you again so much. Did you want to comment on that resource at all?
I think it's a detailed way of putting from step-by-step plan of how you want to do the optimization with all the challenges, the ups and downs that you might face, and how you can find a solution for them. Other than that, thank you so much for having me, and I'm also available via email if anyone has any question or they are doing this similar work. I'm happy to answer questions as well.
Very good. Thank you so much. I do want to thank our audience for joining us today and for their interesting questions. Questions that come in during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Dr. Ahmar, thank you again for your time today. I do want to thank LabRoots. Yes, and I want to thank Leica Biosystems for underwriting today's educational webcast. Today's webcast can be viewed on demand, and LabRoots will alert you when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. Thank you again for joining us. Have a wonderful day, everyone. Bye-bye.